Hello everyone, welcome back to our channel. I'm Dr. Ashmita. Today in this video, we'll study about the signal transduction pathways of non-receptor tyrosine kinases. That is the second one among this chart, non-receptor tyrosine kinases. Let's begin. The non-receptor tyrosine kinases, also abbreviated as the NRTKs, these are cytoplasmic enzymes that have tyrosine kinase activity but do not have their own transmembrane receptor domain. That means we are calling it a non-receptor. That means they are not a receptor by themselves. They are not a receptor by itself, but they are tyrosine kinases. Tyrosine kinases mean these are the enzymes. They are the enzymes with kinase activity. Kinase activity means that will add phosphate to the substances they come in contact with. Here we are saying tyrosine kinase, that means they add phosphate to the tyrosines here. So they are the cytoplasmic enzymes. Kinases means they are cytoplasmic enzymes present in the cytoplasm that have tyrosine kinase, that means they add phosphate to the tyrosine residues and they do not have their own transmembrane receptor domain. We are calling it a non-receptor, so though they don't have the receptor structure. Here, in the first video, we have studied about the receptor tyrosine kinase, that is a receptor by itself. Here, these are the receptor monomers with extracellular domain, transmembrane domain, and the cytosolic domain. The cytosolic domain, they have their own engine of the kinase. So, such is not a case here. These kinases, they don't have a receptor molecule. Where is it? It is present in the cytoplasm, just lying in the cytoplasm. They don't have structure present on the membrane itself. They are unable to receive the signal from the external environment. So they lack this structure. The structure is not present. Only the machine is present within the cytoplasm, that too. So hence they are the cytoplasmic enzymes with tyrosine kinase activity but don't have their own transmembrane receptor domain. So it is not a receptor by itself, hence non-receptor, but they are enzymes with kinase activity, hence tyrosine kinases. Hence the name is justified. And what is the main function of NRTKs? They non-covalently associate with cell surface receptors that lack intrinsic kinase activity. For suppose this is a cell membrane, resting on the cell membrane are these receptors. These are the two monomers of receptors upon ligand binding. So ligand binds to what? To convey some signal to the interior of the cell. Ligand binding is occurring. This receptor dimerization is occurring. Receptor is getting activated, but they don't have few of the receptors. They don't have their own machinery. They don't have their kinase. They don't have their kinase activity. So, to come when the machine is absent, they are able to receive the signal, but this receive is not being able to be transmitted inside. The receive does not reach to the interior. So, this is an helpless receptor. These heroes, the NRTKs, that is the machines, they have the machine but they don't have the structure. So, these machines will go towards the structure to help them. Here the structure, the receptor structure is receiving the signal. They don't have the machine, they are unable to transmit the signal. Now they have the machine, they have the main function is present, but they don't have structure. Now this NRTKs will move towards such helpless receptors. They give their machinery, they give their engine and help these receptors to transmit signal from the external environment towards the interior of the cell. That is the main function of this NRTKs. The NRTKs being the enzymes present in the cytoplasm, but they lack the structure. That is, the structure is absent. So whenever any helpless receptor without a engine is present, these engines move towards that structure and help them to transmit the signal. So this is the main function of non-receptor tyrosine kinases. Let's see the examples of ligands. Example of ligands that act via this NRTKs. We have cytokine, we have antigen, number 3 interleukins, number 4 interferons, number 5 erythropoietin, number 6 thrombopoietin, 7 growth hormone, 8 prolactin, and finally leptin. These are the few examples of ligands that act via this NRTK pathway. So let's see the mechanism of action of these NRTKs. They will study under these headings. First, beginning with ligand binding. A ligand has to bind to the receptor, which is a helpless receptor without a kinase. Now the receptors will undergo dimerization and this JAK activation. These kinases, JAKs here are the tyrosine kinases, Janus kinases. These are the NRTKs here. These JAKs, the NRTKs get activated. They will phosphorylate the receptors tyrosine residues. When they are activated, they will phosphorylate the receptors tyrosine residues and form a phosphotyrosine. Next, for number four is recruitment and phosphorylation of STAT proteins. After the JAKs get activated, they activate the receptors. After that, even the STAT proteins present in the cytoplasm, they also get recruited and phosphorylated. And this STAT dimerization. After activation, the STAT also form dimers. And this nuclear translocation, they are translocated towards the nucleus. Inside the nucleus, this transcriptional activation of target genes. Inside the nucleus, this transcription of certain target genes. Finally, signal termination. After the function is delivered, the signal finally it has to get terminated to prevent overactivation. This will study the mechanism under these headings. Let's begin each one of them in detail. Ligand binding. Here you will take an example of a cytokine. Cytokine is a ligand, or you can also take a growth factor, or erythropoietin, or a thrombopoietin, any of which can be a ligand here. This receptor here is a cytokine receptor. This is a cytokine receptor. So this ligand has now bound. It is binding to the receptors. This is one monomer. This is one monomer. Ligand binding has occurred. Because of ligand binding, there's certain conformational change. That is, the three-dimensional structure of the receptor has undergone change and now they form a dimer. They form a dimer. The receptor has formed a dimer. That means the two receptor monomers now they are adjacent to one another. But there's no kinase in their cytosolic domain. The cytosolic domain, they don't have a kinase. Now, because of this receptor dimerization, JAK molecules, these are the JAKs, these are the enzymes. JAKs means Janus kinase. JAKs means Janus kinase. These are the non-receptor tyrosine kinases present in the cytoplasm just under the cell membrane. This is a cell membrane. So just under the cell membrane, these JAK molecules are present. These molecules, these tyrosine kinases, they get recruited towards the receptor. One JAK molecule will be associated with one monomer. So one Janus kinase will be associated with one receptor monomer. We are calling it a Janus kinase. So it will add phosphate group to one another. This is number one JAK molecule. This is number two JAK molecule. Number one will add phosphate group to the number two. Number two will add to the number one. So I'll give you one, you give me one. Number one JAK has given the phosphate to number two. The number two JAK has given the phosphate to number one. And to whom it is giving? To the tyrosine of the JAK itself. So the JAK molecule has a tyrosine. Each JAK will have a tyrosine residue. So this JAK is phosphorylating this tyrosine present in the number 2. This number 2 is also donating the phosphate to the tyrosine of the number 1. Hence, it is forming a phosphotyrosine here. Now, this is called a transphosphorylation. It is called a transphosphorylation. See, the ligand binding will induce dimerization. Two JAK molecules brought together. A transphosphorylation. This is giving one another is called transphosphorylation. They are giving phosphate to tyrosine residues on the each other. Now this phosphorylated JAKs are called active JAKs. The phosphorylated JAKs thus formed are called the active JAKs. Now this active JAKs, they cause phosphorylation of the receptors tyrosine residues. The JAKs, they'll cause phosphorylation of the receptors. The receptor cytosolic tail will also have a tyrosine residue. Cytosolic tail will also have a tyrosine residue. The receptor has amino acid among which tyrosine residue is one among it. The tyrosine residue present in the cytosolic tail will also be phosphorylated by this JAK. It will also be phosphorylated by this JAK. So the JAK first, it will transphosphorylate each other. After becoming active, it will phosphorylate the receptors tyrosine also. Therefore, the active JAKs phosphorylate tyrosine residues on the receptor's cytoplasmic tail, forming a phosphotyrosine. Finally, a phosphotyrosine is formed. A phosphotyrosine is formed here. Phosphotyrosine is formed on each of these receptor monomers. This phosphotyrosine will have a docking site for SH2 domains. The phosphotyrosines formed, they will act as a docking site, that means a binding site for SH2 domains. SH2 domains. These SH2 domains are present in other certain cytoplasmic proteins. Let's discuss now. So cytoplasmic proteins are the STAT proteins. So the phosphotyrosine, they will act as a binding site for SH2 domains. SH2 domains are present in certain cytoplasmic proteins called the STAT. They have this SH2 domains. This SH2 domains are present on the STAT molecules. This SH2, this STAT molecules will be recruited towards this phosphotyrosine of the receptor. Now, there is recruitment and phosphorylation of the STAT. We have studied the recruitment. The STAT molecules with the SH2 domains in the cytoplasm will bind to the receptor via SH2 domain. Now the JAK, the phosphorylated JAK, it is the JAK is already attached here. This is the JAK. The active JAK is still present. This is the active JAK still present. It has phosphorylated each other. It has also phosphorylated the receptor, and because of this phosphotyrosine, STATs have come here. The STAT molecules, they have come here. STAT molecules. Now the JAK will also phosphorylate the STAT molecules. Here the STAT is present. This JAK will also phosphorylate the STAT molecules. So JAK will phosphorylate STAT on the specific tyrosine residues. Even the STATs will also have a tyrosine. This JAK, active JAK, will phosphorylate the tyrosine of the STAT and form a phosphotyrosine. Now, these phosphorylated stats are also called the active stats. These are the active stats now. The active stat, because it is active, now it will dissociate from here. It will dissociate from the receptor, from the membrane. This will dissociate from the receptor. The stat molecules was bound to the cytosolic tail. It will dissociate. After becoming active, it will dissociate from the receptor and move towards the interior of the cell. And the two stat molecules, here one stat with phosphotyrosine. They have a phosphotyrosine which is bound to SH2 here. It is bound to SH2. They will interact. These both will interact. Because of these interactions, a dimer is formed. Dimer means a couple. Two stat molecules will form a couple. Here there is a dimerized. This is a dimer. Dimer stat is formed. Because of formation of a dimer, there is expression of certain sequences called nuclear localization sequence. There is when dimers phosphorylate stats will dimerize and they form a dimer. The dimer will expose a nuclear localization sequence, NLS, nuclear localization sequence. As the name suggests, it will locate the nucleus. It will get attracted towards the nucleus. So these dimers, they start moving towards the nucleus. They enter the nucleus via certain pores. These are the pores of the nuclear membrane. These dimers will enter through these pores. It will enter the nucleus. It will translocate to nucleus into nucleus through the nuclear pores, and they cause direct gene regulation without secondary messengers. That means, see, fatafat, here the receptor got dimerized, JAK molecules came, JAK transphosphorylated each other, JAK also phosphorylated the receptor, because of this phosphotyrosine of the receptors, STAT molecules got recruited. Now JAK also phosphorylated STAT. Active STATs became a dimer. They got inside directly the STAT molecules went inside the nucleus. So it is like a direct action. STAT molecules from the cytoplasm went there, got active, form a dimer, came inside the nucleus. So there's no mediators in between. There are no secondary messages. It's like a direct path. So it is like a direct gene regulation without secondary messengers. It is a fast and direct path from the membrane towards the nucleus. From here, they went towards the membrane, got active, directly enter the nucleus. So it's a direct pathway. After entering the nucleus, the start dimers will bind to specific DNA response elements in promoters and enhancers. So inside the nucleus, this is a DNA. They'll bind to DNA at specific, they'll bind to the enhancers, they'll bind to the promoters at specific DNA response elements, and they'll recruit certain co-activators. Certain co-activators are recruited, or RNA polymerase 2. They'll also recruit the RNA polymerase 2. Recruit RNA polymerase 2 complexes. And after that, they cause transcription of certain genes. Transcription means this DNA will be converted into mRNA into mRNA. Finally, it will be translated into a desired protein. Depending on the DNA, it will translated into a desired protein. This is about the transcription of certain genes, like transcribes a proliferation genes. It will result in production of protein called cyclin D c-MYC. Cyclin D and c-MYC are involved with cell cycle proliferation. They'll cause the cell to enter into cell cycle. So proliferation. Certain differentiation genes like GATA1 protein involved with cell differentiation. Anti-apoptotic genes that which inhibit cell death like BCL-XL, MCL1. These are the anti-apoptotic proteins which will inhibit cell death. And certain feedback inhibitors also. Feedback inhibitors means that will suppress this own cycle, this own signaling, like SOX. SOX is a suppressor of cytokine signaling. To prevent overactivation, suppressor of cytokine signaling is needed, which is also synthesized because of the cell signaling only. So cell signaling has transmitted here, they'll transcribe certain gene which is required for growth, which is required differentiation, which is required for anti-apoptosis to inhibit cell death, and also to stop the signal. To stop the signal also, it will produce certain proteins like SOX. This is about the transcription of target genes. Finally, now the desired action is given of either that growth or a differentiation or the anti-apoptosis or the synthesis of inhibitors also. The final action is given. Now this signal for coming from the receptor, from the ligand has to get terminated. How it is terminated? In the of the step 6 itself, SOX proteins, that is this one, the suppressor of cytokine signaling. These SOX proteins will bind to JAKs and the receptors. This SOX produced here, SOX proteins, they will bind to the receptor. They'll bind to the receptor, or they'll bind to the JAK itself, JAK molecules, and directly it will cause inhibition. Or there are certain phosphatases, like proteins called protein tyrosine phosphatases. As the name suggests, phosphatase, it will mean breaking the phosphate. It will remove the phosphate. It will dephosphorylate. It will remove the phosphate from JAKs and STATs. Here the JAK active STAT, they are causing the cell signaling. So they'll remove the phosphate from here. They'll remove the phosphate from JAK means inactive JAK. Removal from the STAT means inactive STAT. There's no cell signaling happening. So this protein tyrosine phosphatases will dephosphorylate, remove the phosphate from JAKs and STATs. And thirdly, this receptor internalization and degradation. Internalization means whenever such receptors are exposed on the cell surface only, then only they are able to receive the signal. So don't make them exposed. Don't expose the receptor. They'll internalize. The membrane will internalize. It will take the receptor in. It is not exposed on the surface. Therefore, they cannot receive the signal. Or the entire receptor itself is degraded. When there's no receptor, there's no ligand binding, no signal. So, in these three pathways, the signal termination occurs by the SOX proteins transcribed from the nucleus, or the protein tyrosine phosphatases that which remove the phosphate from JAKs and STATs, or the receptor internalization, don't expose them or degrade them. This will help in to avoid hyperproliferation. Here, because of this signaling only, there's proliferation happening or differentiation happening. To prevent hyperproliferation, to prevent cancers also, we have to terminate the signal, or to prevent the inflammation, because it is a cytokine signaling happening. Means inflammation. To prevent over inflammation also, signal termination is a must and must. So this is about the mechanism of action of the non-receptor tyrosine kinases. Let's see few types of NRTKs. Like there are many families of NRTKs. The most important among them are the JAK family, SRC family, ABL family, FAK family, TEC family, SYK or ZAP70 family, CSK family, FER family. These are the few families of NRTKs. Each of these families have members, among which we'll take up a few important members that have clinical correlation. Among the JAK family, there are JAK1, JAK2, JAK3, and TYK2. These are the few members of the JAK family. Here, JAK2 mutation is associated with myeloproliferative neoplasms like polycythemia vera or essential thrombocythemia. Polycythemia vera, essential thrombocythemia. And JAK3 mutation is associated with severe combined immunodeficiency. Coming to the SRC family, this SRC protein overactivation will lead to cancers of breast, colon, lung. ABL family, we all know that CABL, the classical, the BCR-ABL fusion seen in translocation 922 will lead to chronic myeloid leukemia. And coming to this FAK family, focal adhesion kinases, this overexpression is associated with metastatic cancer, especially of breast and colon. TEC family, this Bruton tyrosine kinase (BTK) is a Bruton tyrosine kinase mutation associated with X-linked A-gamma globulinemia. Coming to the ZAP70 family, here we'll take this ZAP70. It has two forms, deficient form and overexpressed. Deficiency will lead to severe combined immunodeficiency because there's no T cell receptor signaling. It is deficient means no T cell receptor signaling. If it's overexpressed, there's increased survival of B cells. That means it will lead to chronic lymphocytic leukemia. Chronic lymphocytic leukemia. And coming to the CSK family, whenever CSK is lost, SRC will be hyperactivated. The normal function of the CSK is to inhibit. It is to inhibit SRC. When the CRK is lost, the inhibition is lost, SRC will be hyperactivated. What will happen when SRC is hyperactivated? It will result in cancers of breast, colon, lung. So it will result in the cancers of breast, colon, and lung. Coming to the FER family, that is, FER will normally regulate the cytoskeleton of cells. When it is overly expressed, there is increased motility of the cells. That means it will favor tumor invasion and metastasis. This is about a few clinical relevant mutations. We'll consider one important family and one important member for the clinical understanding. So we'll take an example of JAK2 molecules, the JAK2 proteins, that is Janus kinase 2. So they are Janus kinase, they are associated with erythropoietin signaling. So we'll take an example of erythropoietin ligand here. It will bind to the cytokine receptor. It is an hematopoietic cytokine because it will induce cell signaling. Therefore, hematopoietic cytokine will bind. It will act as cytokine receptors. So normally, whenever erythropoietin is bound to cytokine receptor without kinase domains, the receptor will same mechanism. The receptor will dimerize, but they don't have kinase. So they will recruit JAK2 from this under the membrane. They will recruit the JAK2. The JAK2 will bind to the receptors, the cytosolic tail, transphosphorylate each other, also phosphorylate the receptors, and they will attract the STAT molecules. STAT also get activated. They'll move towards the nucleus, cause proliferation of hematopoietic stem cells. Since it is erythropoietin, they'll cause increased production of this hematopoietic cells. So this is the normal function of this erythropoietin and acting via JAK2. So JAK2 here normally it has like many homologies which are called Janus homology. It ranges from JH1 to JH7, among which we'll take two examples. These two are considered very important. JH1 is called the kinase domain. It is an active domain. JH2 is a pseudokinase. The function of this active domain or the kinase domain is to phosphorylate STAT. We are saying here JAK2 has been recruited. It will phosphorylate each other. It will phosphorylate receptor. It will phosphorylate the STAT also. How it is phosphorylating? By this JH1. JAK2 has this JH1. JAKs have this JH1. This JH1 is responsible for this phosphorylating function. Phosphorylating function is because of JH1. JH2 is a pseudokinase that will inhibit, they have an inhibitory effect on JH1. So, whenever there's a receptor signaling, whenever erythropoietin is binding to the receptor, there's receptor signaling occurs, the JH2 will be inhibited. This JH2 inhibited, the inhibitory control lost. JH1 is now free to act. So JH1 is free to act. What is the normal function of JH1? To phosphorylate the STAT and activate the STAT, cause nuclear translocation and transcription of genes. This is the normal JAK2 function. Whenever there is a JAK2 mutation, JAK2 mutation here it is a V617F mutation. That means V means valine getting replaced by phenylalanine. Valine here Valine, it is getting replaced by phenylalanine at codon number 617, hence called JAK2 V617F mutation. Whenever this mutation is happening, this JH2 is permanently inhibited. JH2 is permanently inhibited. That means JH1 is continuously active. This continuously active JH1 means continuously they stimulate the STATs. Continuously, they will stimulate the STATs. The STAT dimers will continuously activate. They'll translocate to the nucleus, they cause continuous proliferation, continuous signaling. So therefore, it is a ligand independent. Even in mutations, even in the absence of erythropoietin, the absence of ligand, there is no need of ligand. Even in the absence, in cases of mutation, I'm saying, even in the absence of a ligand, there is proliferation and survival of hematopoietic cells because JAK2 is mutated, JH2 is permanently inactivated, JH1 is acting, there is causing continuous activation of STAT, continuous signaling. So, continuous proliferation and survival of hematopoietic cells that will result in polycythemia vera, essential thrombocythemia, thrombocythemia, and thirdly PMF, that is the primary myelofibrosis, and rarely does it cause acute lymphoblastic leukemia ALL or atypical CML, that is atypical chronic myeloid leukemia. These are the commonest cancers caused by JAK2 mutations. This is one of the examples, important example of a JAK2 mutation. So this is about the non-receptor tyrosine kinases. We have studied the mechanism, what is it, the introduction, the definition, the examples of ligands, mechanism, how do they, like Janus kinases, that means how they come in to the help of, to the rescue of these receptors without kinase, without the machinery, and how do they activate itself, the receptor, the STATs, getting directly transmitted to the nucleus, direct action on the nucleus, transcribing many genes. Whenever there's a mutation, this continuous signaling leading to such cancers. This is about the non-receptor tyrosine kinases. So if you find this video useful, please like, share and subscribe to our channel. Thank you.